we're joined by two wonderful speakers today, Dr. Derek Bowie and Dr. Karen Zito. I'll introduce Dr. Bowie first, and then we'll move to Dr. Zito, and there'll be time at the end for questions. So if any questions come up, feel free to put them in the chat, and we'll have an open question period at the end. So Dr. Derek Bowie is a professor in the Department of Pharmacology and Therapeutics at McGill University, and he's also the co-director of Cell Information Systems. So Dr. Bowie, if you want to share your screen and take it away.
Okay, so maybe I could just start by thanking Keith MacArthur, all the Cure Green people who are involved in putting this whole thing together, and of course Quinn, who's been really essential for kind of getting this session together. What I thought I would do, because I was asked to talk about rethinking ionotropic glutamate receptors, and I thought I would tell you a little bit about how we're thinking about it. It's a little bit different from how other people are thinking about it, and I think that's important for us to all have a slightly different perspective on things. So I wanted to tell you about that, and I guess the interesting thing is it's definitely starting to give us insight into greed disorders in a way that maybe wasn't immediately anticipated. But the other thing is, and maybe if I can just pull up my, can you see my laser pointer, everybody? It's actually bringing out new biology about some of these proteins we've been looking at. So in a funny kind of way, studying greed disorders is actually giving us insight into our understanding of different types of ionotropic glutamate receptors. So what I thought I'd do is just give you a general perspective on things. And I guess where that really comes from is at the end of last year, I was asked to write an article for this magazine called The Scientist. And they weren't completely clear what they wanted me to talk about. They suggested a few things. And I had just been at the GRI Europe conference in Milan, and also just been in Toronto. This was in the late fall, kind of the winter period of 2025. And I was at a Fragile X conference, which my lab also works on, which is another intellectual disability disorder. And so what I thought I'd do is talk about glutamatergic synapses and something that our lab is starting to really feel is a very important thing, which is that they're really a hub for intellectual disability disorders. And so rather than writing a very detailed scientific article for the magazine, what I thought I'd do is write it. I would write it for three kinds of persons. One was so surprising, but one was for the politicians, because politicians don't know all the details about intellectual disability, but they're very sensitive to it and they want to do something about it. But you have to give them something that they can get behind. Also wanted to write the article also for people in industry, because they too are would love to develop treatments for intellectual disability, but are in many ways, and I'll explain it in a few seconds, are kind of struggling to try to think about a kind of common strategy. And I thought, well, if we sort of say to people, really think about trying to focus on glutamatergic synapses, that may be one to do it. And then finally, I thought I'd sort of share our kind of how we're looking at things from a scientist perspective. So in terms of the politicians, I was just recently in Ottawa, which is where the Canadian Parliament was, and I met with a bunch of politicians. Here's one of the pictures from that time. And I spent the whole day there, so I really had a lot of meetings, even with Health Canada. And the situation in Canada is unfortunately not as good as it should be. There's a bit of an existential threat to funding here. So if you just look at the graph of funding over the last 20 odd years or so, it's gone down by half. So all the research that we would love to do on greed disorders and any other kind of CNS disorders is dropped by half. So that means, once that happens, then really good science is not getting funded. So we were there primarily to talk about that and also get a sense of how politicians view things. And as I said, I think they're sympathetic, but they want to get behind something big. They won't want to get behind just Fragile X or greed disorders. They want to actually get behind a whole bunch of things, maybe intellectual disability. And so that was a very sort of important message that kind of came to them. In terms of industry, I actually a few years ago co-founded this company called Pharma, and we're in the process of bringing forward a treatment for Fragile X syndrome. And in that process, you have to go out and raise a lot of money. You have to raise millions of dollars. And again, you meet all these business savvy entrepreneurs, mainly in the US, actually raising a lot of money in the US. And They're again, they completely appeals to them about the idea of the disorders and things like that. But what they'd like to do, because they find themselves doing due diligence on everything that turns up across their table that they're thinking of investing in, is that they're sort of always telling us, I wish people would come up with some sort of one common thing that they think needs to get fixed in intellectual disability. We can go after that and we can get funding behind that. So if one's thinking about intellectual disability, and if you're trying to put them all together as a kind of strategy to try and get funding from a political class and all from the industry, then we know that glutamate synapses are actually packed full of different kinds of proteins. So how many proteins we're really talking about? And so we're talking about maybe hundreds or thousands of proteins. So here's just some of the proteins that we know that are in glutamatergic synapses. We know that many of these, whether truncations or missense mutations, all cause intellectual disability. The ones, of course, that we know something about, which maybe is a little bit outside the GRI field, but there's FMRP, which is tied into Fragile X syndrome, the SYNGAP1, which is an important protein that sits just under synapses, which causes deficits in that cause SYNGAP1 related intellectual disability, and the SHANK3, which is tied into Phelan-McDermid syndrome. So from our perspective, from in my lab, all of these proteins and the ion channels that we all care about, so the AMPA receptors, the KNUT receptors that some people on the call here will be thinking about, and also, AMPA, NMDA, and KNUT receptors, which you'll be hearing about perhaps later on this afternoon. All of these, we kind of think of them as all falling in together into common deficits, different origins, but they all commonly lead to intellectual disability. And you can go beyond that, actually. So if you start to look at the cell adhesion molecules, so here's a bunch of them that we've sort of put together in this review just to try and talk about it. So that probably the most commonly cited one is the neurexin-neuroligin interaction that help form the specificity of synapses, glutamatergic and GABAergic synapses. And missense mutations in these proteins also cause intellectual disability. So in our mind, in a sense, to try to broaden this and bring it as a bigger appeal to industry and also as a bigger appeal to the political class, is that we think we should try and really think about these as just one disease with different kind of factions that we have to solve. And I've got colleagues in cancer, and there's many different kinds of cancers, and what they do is they band together to try and argue in favor of let's solve cancer. They don't worry about telling all the details of that. So something that's really been an important thing for us that we've sort of scratched our head about is work that's come out of a number of people who are on this call right now. So Steve Janellis and Casper Hans and various other people have been looking at, Ian Combs as well, who's been looking at all the missense mutations that are found in AMPA receptors, in addition to all the clinical researchers that have contributed greatly to that. And if you look at AMPA receptors, so that's just one of the proteins that we're interested in, but there's now probably over maybe 100 missense mutations found in AMPA receptors. There's 4 genes that encode AMPA receptor subunits. And you can see the different proportions that are found here. And what's remarkable about when you make this plot is it's not immediately obvious why AMPA receptors would cause intellectual disability. You can kind of hand wave a little bit and sort of say, well, they're really important for glutamatergic transmission. And if you mess them up, then that's going to mess up transmission. But other than that, we don't really have a very, very strong explanation to why AMPA receptors in particular, why they should cause intellectual disability and why it's so profound. And so what you could do is you could see yourself 100 missense mutations. Maybe you could go off and study all of them one by one and classifying them into loss of function, gain of function, and things like that. But we think that's going to be a very, very challenging thing. Not only is that a lot of work in itself to try to do that, but the other thing is that AMPA receptors, unlike NMDA receptors, actually don't sit in our glutamatergic synapses by themselves. They actually sit with a whole bunch of different types of auxiliary subunits. And so over the years, work from our lab and other labs, structural biology, proteomics labs, etc., have revealed there's a whole bunch of different types of auxiliary proteins. I'm not going into great detail because the experts on this call know what I'm talking about, but there's the Claudin superfamily, the TARPs, and there's also GH2NL. TARPs kind of dominate AMPA receptors. So it looks like every single AMPA receptor just about when it assembles in the brain, actually has a TARP bound to it. There's about a smaller percentage, 5%, that we had a paper with Bernd Fackler showing about 5% are probably get GS21L. But in the forebrain, TARPs also team up with cornichons with AMPA receptors, and that seems to be very important. And they also team up with the shishas or the C-camps, depends what you want to call it, and also SINDIG, which is work done by Elva Diaz out in UC Davis, where Karen actually is. I think she's probably part of the same department. And there's a whole bunch of rules in how these AMPA receptors come together. So if you want to start to study all these 100 missense mutations and then classify them, then you also have to look at them like this. The other added complication, of course, is that the regulation of these receptors is very sensitive to the development stage of the brain. So here's just a snapshot of RG editing site, which I'm not going to talk about, but it turns out it's actually a very important thing. Alternate splicing, which we've talked about before, because it switches on and off the effect of TARPs. And also claudin expression. You can see that all of them change very dramatically during brain development. The alternate splicing is very interesting, because it looks like everything's flip, and then it becomes a mixture of flip and flop. And we haven't published this yet, but there's evidence, we get pretty strong evidence that different brain regions have different levels of flip and flop, and so that's very dynamic, and so that means if you want to really study the AMPA receptor, then you can't ignore the fact that it's got all these different aspects of regulation which are changing based on development, brain region, the cell type, and even synapse. And so how can you try and get to that? So the way we started to think about this, and it would be surprising to everybody at the time, but we said, well, why don't we go back and just think about how did AMPA receptors come about? Let's think about them in terms of evolution. And so here's a kind of schematic that I put together for a recent article, an opinion article I published in TINS. And what I did is just something very simple. I just looked at the different types of AMPA receptor subunits that you could get in different kind of species. So these are the vertebrates, these are the invertebrates, so protostomes. So they kind of went a different direction in terms of evolution. And then there's these non-bilaterians. So these are the very earliest organisms that were around about 650, 700 million years ago. So a very, very long time. And it turns out there's AMPA receptor-like. They're not AMPA receptors, but there's AMPA receptor-like proteins in these very, very early organisms, and they can transport calcium. Actually, it's amazing, right? They all transport calcium. All the elements that you need in a modern AMPA receptor to transport calcium are found in these organisms that were found 700 million years ago. So it's something which was discovered a long time ago, right? But what's different is that these regulatory features of AMPA receptors, they only turned up in vertebrates. They only turned up in early vertebrates, in fact. And that's also true for the auxiliary subunits. Although there's some cornichons early on, they're not really involved with AMPA receptors, doing other cell biology things. And although the invertebrates do have some auxiliary proteins, they don't get regulated in the same kind of way because they do not have RNA editing and alternate splicing, which has gone on there. So that's an important thing. And then I guess the really important thing that made us really revisit the whole AMPA, it's one of the sort of very important features of AMPA receptors, is that when we actually kind of looked at the features, the genomic sequences, and the properties of these receptors, what we found is that when you get into the early invertebrates, the first thing that really appears in the jawless vertebrates, so these are the lampreys and hagfish, so these are very kind of ancient early vertebrates, they actually have alternate splicing, but they don't have RNA editing. They do have an arginine in the AMPA receptor subunits, but that's because it's genomic. It's not there because of RNA editing. RNA editing came about a little bit later, actually, in vertebrate evolution. And what that says to us is that actually all the AMPA receptors we have in our brain, which for the last 30 years, ever since they've been cloned, we've tended to think that they're really just calcium impermeable. We thought, well, maybe actually what's been going on for the last hundreds of millions of years is that all the AMPA receptors and ancient AMPA receptors were all very highly calcium permeable, just like NMD receptors. But because of the developing, an emerging and evolving brain that requires a high degree of complexity, then that calcium permeability was tuned down. And so what we thought is that maybe there's AMPA receptors out there that have the potential to be calcium permeable. And so we went and looked at that. And so the conventional view that we think about with AMPA receptors is that, so it's kind of described here in the first figure that we had in our paper, is that most of the AMPA receptors in the brain are like this Purkinje cell, which is shown here. I won't go into how you do these experiments, but you can basically measure the relative calcium permeability with respect to sodium, and it's got pretty low calcium permeability, these Purkinje cells. And then there's a few cells, not many actually, but one good example is a Bergman glia that doesn't express the GLUA2 subunit, and they're very, very highly calcium permeable. But there's not that many cells that have these. And so we tend to think that kind of most of the AMPA receptors in the brain are calcium impermeable. But we thought that what's actually happened is that during evolution, the calcium permeability is there. It's just that it's been features that have been built in, so RNA editing, the expression of auxiliary subunits, that haven't completely got rid of calcium permeability, but they've shut it down, because if you get too much calcium permeability, then you get neurotoxic effects within the brain, and that's been known for some time. Okay. So to try to look at that, I'm not going to show you the data just because I'm supposed to speak only for 20 minutes. We looked at AMPA receptors. We assembled AMPA receptors to be found in the forebrain, so the cortex, hippocampus, but also in the cerebellum. The cerebellum is mainly TARP-dominated AMPA receptors, whereas in the forebrain, so the hippocampus, the cerebral cortex, it not only has TARPs, but has the cornichons, but also has different kinds of shishas and syndics and things like that. So we looked at them, and what we found is rather than having just two populations of different kinds of AMPA receptors, the majority would be calcium impermeable, and then a couple of them being calcium permeable. What we actually found is that there was a continuum of this expression of AMPA receptors. And so that came as a surprise, I think, to a lot of people. It didn't really come as a surprise to us because we had hints of that already when we were looking in the retina, and we'd never published it, but we'd actually seen some evidence for that also in the hippocampus. Now, what you can see is there's a continuum of that. But what's interesting about it is that the naturally occurring AMPA receptors, there seems to be this kind of borderline where they can get more calcium permeable than sodium ions, right? So they're about the ability of calcium to transport the pore is just about as good as sodium, whereas in Bergman glia, for example, which is shown here, they're about 3 or 4 times better at transporting calcium than sodium. So there is a limit to which a naturally occurring GLUE2 containing AMPA receptor seems to transport calcium. And then the other thing is, of course, that it doesn't let the pore properties don't change that much, so you can let polyamines there block. Now, why am I telling you about that at a GRI conference? And the reason that was important is we actually started to look at some missense mutations that were found in some kids with GRI disorders, and I'm going to show you a picture of those kids in a minute. And what we found is that what happened to them is that they now had much larger increases in calcium transport, and they also had sensitivity to polyamine block, even though they had that GLUE2 subunit. And so the kids that I'm talking about is this kid here, this young girl here, patient 7. This is published in 2019, so she's probably into her teens now. And this little boy here, patient 3, and there was another patient as well. And all of them, based on the experiments we have done, will have higher calcium permeability. And they kind of get over that borderline that we think is a strict line, that once you go over that, then the calcium starts to be detrimental to the developing neuron. So is that a reasonable way to think about many of the other missense mutations that Steve and Casper and all these colleagues of mine over the years have been working away and mapping out? And we think it might be the case, actually. So if you go in and look at what's been put into these databases, what you see is that the pore region is a bit of a hotspot for missense mutations. There's other residues as well that you can find in TM4, which we think are going to be interesting as well. But what it's really saying is the way we think about it from an evolutionary perspective is the way I guess people in green conditions kind of think of these as being these very detrimental mutations. But the way we think about it in terms of this evo-devo perspective that we're taking, so evolutionary molecular biology, developmental biology perspective that we're taking, is this is how evolution tests out things. So the missense mutations that occur, and they're usually de novo anyway, are really how AMPA receptors evolved. And so sometimes it works in your favor. And we think that's how you ended up with the RG site and the QRN site. So it worked, sometimes it didn't. And so you ended up getting favorable things. And the challenge now is with all these kids who are troubled with different aspects and terrible aspects of GRI disorders, is that we're still stuck with that evolutionary mechanism that goes in and looks at very sensitive parts of the protein, or it doesn't look, it sounds like it's active. I'm not suggesting it's active, but the sensitive parts of the protein, if you mutate them, then they've dramatic effects to the protein that in evolution has been beneficial sometimes, but of course, right now, in terms of the kids that we care about, has very detrimental effects. And so that's been a really interesting perspective for us to take, because it's led us into other things that we haven't published yet, but it kind of fits into that mold. And I thought that was an interesting way to think about it, because it's just another angle. And so it's also good for people then to start to think about it and even criticize it. But at least it will lead to debate. So I wanted to use my 20 minutes, which is now up, just to explain that. So just before I finish, I just want to thank all the people that's been involved in putting a lot of this work together. I want to thank our funding. We've also got funding from GRI, so I'm really appreciative of that. And once Karen's presentation is finished, I'll be happy to take questions and discuss whatever people are interested in chatting about. So thanks again.
Awesome. Thank you so much. That was wonderful. So next we'll have Dr. Zito, and then, like I mentioned, we'll have an opportunity for questions at the end. So again, if any come up, just feel free to put them in the chat, but there will be an opportunity later. So Dr. Karen Zito is a professor of neurobiology, physiology, and behavior in the Center for Neuroscience at the University of California, Davis, where she investigates how neural circuit connections are modified during learning and how they're affected by diseases that disrupt learning.
I'd like to start by thanking the organizers and especially Quinn for including me. I'm relatively new compared to the other scientists here to GRIN research. And I've learned so much in preparing, and also just appreciated hearing from the families this morning. It was really insightful. So like Quinn said, I'm at UC Davis. Oh, let's see if I can get it. There we go. And my lab has been studying for almost 20 years now how the brain changes when we learn. And in particular, we're interested in identifying the molecular signaling pathways that support learning and the cellular changes in the brain that support learning and how they're affected by disorders that disrupt learning. And recently, this has led us down the path of looking closely at a novel signaling pathway downstream of the NMDA receptor, and that's what I'm going to talk to you about today. So I just wanted to orient you first a little bit on essentially the what types of changes do happen when we're thinking about cells in the brain during learning. And what we can think about is that these cells are changing both in the strength of their connections. So our neurons are connected into circuits, and how strong they connect can change. But also, there's a lot of structural changes in the brain that support those functional changes. So neurons and glia change in their structure to solidify those functional changes. And these changes in brain structure are thought to be really crucial for the stabilization of the learning associated changes in synaptic strength. And so we've been focusing in on these structural changes and how they're coordinated with functional changes in the brain, and most recently, how the NMDA receptor is involved in that. So just to give you an example of the type of structural change we look at these really small protrusions that come off of neurons. They're called dendritic spines. You might have heard of them. One example here is when a mouse is learning, we can image inside the mouse brain if its neurons are labeled fluorescently. And when a mouse is learning a task like rotating balancing on a rotorod, we can image before and after it learns. And what we can see is that there's an increase in spine growth during learning. And that increase in spine growth, the amount of it is associated with performance, suggesting that we're watching these new circuits form as the animal is learning. And this has been shown in a really large number of labs. I can't list them all, in organisms as varied as mice and songbirds and monkeys. But if we can also see that during learning, it's not just the growth of these new synaptic connections, but the brain adjusts by removing ones that are unwanted. And so again, we see there's a really strong correlation of elimination of synapses with performance as well. So it's this balance of new synapse growth and synapse retraction. And in fact, if one looks at just the change of synapses in an organism of a human over a lifetime, we see that we get these increase in spine synapse numbers robustly during early development. They get pruned back and eliminated as the nervous system is developing and matching to the sensory environment. And then in adulthood, we have this balance. We have new growth during learning and removal of ones that are unnecessary. And with the emergence of symptoms in many diseases that disrupt learning, this is just an example of schizophrenia, we see a disruption in these dendritic spine synapses. And so here in early adolescence, we see them being disrupted in schizophrenia, or here in Alzheimer's disease, very late in adulthood. And there are associated changes in synaptic structure also associated with grin disorders, but it's been, I would say, less well studied than the functional changes so to date. So the NMDA receptor is really important in coordinating these changes in synaptic structure and function, which is what we're interested in, again, because of that stabilization role. So as a new synapse grows, we have the growth of the cytoskeleton that supports the growth of the synapse. And then we also have insertion of AMPA receptors that were so nicely introduced by Derek and effectively in the last talk. And so this coordinated growth and then shrinkage is associated with learning. And again, it's downstream, just one example, it's downstream of the NMDA receptor, and there's downstream synaptic signaling molecules that are thought to support both the functional changes and the structural changes. So essentially the pathway starts, glutamate gets exposed to the synapse gets exposed to glutamate, and depending upon the pattern of that glutamatergic activation, we have, say, the activation of phosphatases that then split to a pathway that leads to synaptic weakening and one that leads to spine shrinkage. So just to go back and introduce the NMDA receptor a little bit more thoroughly, just the idea of what all it takes for the NMDA receptor to function. So the NMDA receptor is a tetrameric receptor. It has two gluon 2 subunits that bind glutamate, so that's shown here, the red binding, and then two obligatory gluon 1 subunits that bind coagonists, which is glycine or D-serine. And so it's just important to note that you need both of these binding for the receptor to open and form a channel. That's the I in GRI, glutamate receptor ionotropic, so channel. It's an ionotropic, lets ions through. The other interesting thing that I want to point out is that the NMDA receptor is blocked by magnesium. So right now this receptor is open, but when it's at rest, it has a magnesium in the pore that blocks the ion flow. And so both of these are going to contribute to what I tell you about in just a minute. And then once it opens, it's a non-selective cation channel, and its special properties of promoting this type of brain plasticity were always attributed to the calcium influx through the channel, which now Derek is showing there's a lot going on with AMPA receptors as well, but this is the traditional way the NMDA receptor was proposed to drive synaptic plasticity. And then over the years, now it's been, several, like almost 2 decades, it's been, evidence has been accumulating the NMDA receptor can signal in the absence of ion flux. And so most of these studies were shown with using various pharmacological agents that can block various aspects of the NMDA receptor. So a glutamate that binding can be blocked with CPP or AP5. The pore can be blocked with MK801, or the coagonist binding can be blocked with 7CK or L689. I'm going to be talking a lot about this coagonist block, so keep that in mind coming up. And if these are blocked, there's a lot of things that still happen that are NMDA receptor dependent. And this is just some examples of endocytosis, excitotoxicity, synapse to nucleus signaling, astrocytic calcium, blood-brain barrier permeability. And what I'm going to focus on today is synaptic plasticity and transmission. And it was really work in the Malino lab from about 10 years ago, a little over 10 years ago, that started us down this pathway and revived this idea that the NMDA receptor can drive plasticity without flowing ions. And that's why I guess I'm in this session, which is to talk about this a non-canonical, this unusual signaling mode of the NMDA receptor. So Ivar Stein in my lab decided he wanted to have a look to see, well, it had been shown by the Malinois lab to be involved in the synaptic weakening, the functional changes. So what about the matching structural changes? And he wanted to look at individual spines. So these synapses that I showed you are so important for driving learning. And so these are just some of the details for the experts on what conditions we're using for our experiments. But here we're going to use one of these drugs. We'll be using both 7-CK and L689 that blocks the coagnous binding that keeps the channel from opening. And I just want to prove to you that this is happening. So we're recording from a neuron in a brain slice, and we're uncaging some glutamate right next to a synapse. And what we show is that, yeah, if we add 7-CK, the current that normally goes through this channel in this spine does not go through anymore. And we can also image calcium, and we show the calcium that normally goes through also does not go through. And so what Ivar went out to test, and this is just one example of the types of experiments we do, is he's now looking at these spines and he's time-lapse imaging, and he's giving a plasticity-inducing stimulus that causes spines to shrink. And this is via the NMDA receptor. And then he's putting on this drug that blocks ion flux through the NMDA receptor by blocking the coagnous binding. And he's showing that they still shrink. So, however, when he puts on CPP, which blocks glutamate binding to the NMDA receptor, they don't. So this is just a quantification. You can see without any drugs, this uncaging of glutamate leads to spine shrinkage. If we block the coagnous, which I just showed you, blocks all ion flux, we see a beautiful shrinkage that's glutamate induced, because if we block glutamate binding to the NMDA receptor, we don't see any change. So we did a bunch of tests. I'm not going to have time, just like Derek said, to go through. I'll just show you a few details. One is that if we block AMPA receptors and group 1 mGluers, we still see this shrinkage. So you can see here, this is blocking AMPA receptors, and then actually I'm getting blocked over here. But if you go one more over, you can block group 1 mGluers and you still see the shrinkage. So it's definitely NMDA receptor dependent, and it's not due to other glutamate receptors. And then Ivar just showed he did this exact same experiment now looking at functional electrophysiological recordings, and he showed that indeed in the presence of L689, he sees an LTD in these synapses that are stimulated by glutamate, even though there's no current flowing through the NMDA receptor at that time. So just to summarize a lot of work, so Ivar started looking at the signaling pathways downstream. And so here what we're arguing is that glutamate binding to the NMDA receptor in the absence of ion flow is going to drive both synaptic weakening, or LTD, and spine shrinkage. And we started putting together this pathway by using pharmacological drugs, by using genetic manipulations, peptide interruptions, various manipulations, and we started putting together the pathway that leads to this plasticity. And then one other study, I'm not going to have time because I'd like to focus on the implications for disease to describe, is that, so what we think this pathway is doing is it's leading down here to disruption of cofilin, which drives changes in the cytoskeleton. So that cytoskeleton I showed you that holds the synapses out there to make them stable during learning, when we have this pathway, it loosens up the cytoskeleton to make the synapse able to be more plastic. And then if it keeps going, it causes the synapse to shrink and go away. In the case, we have a whole nother paper, again, I won't have time to go into, but that looks at how is this pathway involved in the learning-induced growth. And it turns out that during learning-induced growth, we do need calcium through the NMDA receptor to drive spine-induced growth and insertion of AMPA receptors during learning. However, it turns out we also need this ion flux independent pathway. And we showed this by blocking a whole bunch of proteins that are on this pathway. And we think we need this pathway in order to, again, loosen up the cytoskeleton to get the synapse ready for plasticity. And so if you want to read about that, that's already published as well. So what I'd like to focus on is, number one, when do we expect this pathway to be involved in just physiological situations, not using these drugs that are, exogenous, and then how might they be affected by mutations in the NMDA receptor? And so one example of when we would expect this to happen is I showed you that the NMDA receptor is blocked by magnesium at rest, or I told you. And so at single synapses, we have these NMDA receptors that are sitting around blocked by magnesium, and when glutamate binds, that isn't usually enough at one synapse to bring the magnesium block out. So if there's uncorrelated activity at single synapses, we will get this glutamate-induced shrinkage pathway, or synapses lack AMPA receptors. So here's just an example. Here now we have, we're doing single spine uncaging now in the presence of magnesium. And if magnesium is in there at physiological levels, if we just have a single synapse activated, nothing else, we see this spine shrinkage. Mutations in AMPA receptors that would cause synapses to be less effective would be expected again to drive this pathway. And mutations in NMDA receptors that impact perhaps the magnesium block, making it stronger, would also block this, be expected to induce this type of change. Another type of change that appears to be physiologically common is changes in the modulation of levels of D-serine. So for instance, in sleep or disease states, D-serine is modulated. It can be low during rest and high during wakefulness, and this would be swaying the NMDA receptor from going without D-serine ion flux independent with D-serine ion flux dependent. It's also been reported to change during aging in several diseases, including schizophrenia. And so we can show that just by taking a normal slice, nothing changed, no drugs, and then we add a drug that'll eat up the D-serine. And when we stimulate, that'll cause that in a way that doesn't cause any changes at the synapse. Now, if we eat up the D-serine, it causes shrinkage and loss. So again, we can modulate the D-serine levels and show that affects the stability of the synapse. So GRIN mutations that affect coagonist or D-serine affinity would be expected to, again, destabilize these synapses and cause this to happen. And so one more set of experiments that I just have a couple slides on that we did was, like I mentioned, it has been shown that lower D-serine effects is happening in schizophrenia. So in humans, cerebral spinal fluid, we see lower D-serine levels and lowered levels of the activity of the enzyme that produces D-serine. And patients with schizophrenia have lower levels of dendritic spines. And so there's a mouse model that we adopted that was developed by Joe Coyle that shows, again, by interrupting the gene, this serine racemase gene, that there's less spines and behavioral phenotypes that are reminiscent of schizophrenia. So this is just one aspect, modeling one aspect. And so again, our proposal for these situations is that, again, normal signaling where both glutamate and D-serine are bound and the magnesium popped out, calcium can come through and we get spine growth and stabilization. But with reduced D-serine levels, calcium influx is interrupted, and that instead leads the exact same stimulus to drive spine shrinkage and destabilization, leading to spine loss. And I just have two slides showing that indeed in the serine racemase knockout, a stimulus that usually leads to growth, long-term growth, doesn't in the knockout. And we can rescue that by adding D-serine back. So it's not some long-term synaptic wiring change. And that, yes, indeed, there's a shift from in the serine racemase knockout from the wild type that continues spine shrinkage for longer, so that they're predisposed to shrink for more types of stimuli. So they have a disposition to shrink. So just last slide to just say again, in the wild type animals, you'd expect to see both non-inotropic and calcium signaling to lead to spine growth. But when the D-serine levels are changed, that's going to increase the ion flux independent signaling and decrease the calcium signal, leading to spine shrinkage and elimination. So to summarize what I've told you today, NMDA receptor-dependent spine synaptic weakening can be induced by glutamate binding in the absence of ion flow. So this is an ion flux independent way the NMDA receptor can promote the loss of synapses during plasticity. And enhanced ion flux independent signaling likely drives the spine loss in physiological and disease states which are associated with persistent magnesium block or lower D-serine levels. And then mutations in GRIN and GRIA could tune the balance between these two different signaling pathways and altering the brain plasticity that supports learning. And I thought I put, oh, there they go, they're just showing up. So for instance, mutations again that would reduce the affinity or of D-serine or glycine binding, coagonist binding would be expected to do this. Mutations that would increase, say, the affinity of magnesium or it's sticking it in the pore. And then none of the crystallographic structures that you are being shown have this C-terminal domain on them. But yeah, anything that talks between the C-terminal domain and those downstream signaling pathways that I showed you would also be expected to influence dramatically. And in fact, the lab that Quinn came from has a beautiful study just showing that indeed this ion flux independent signaling is driven by a small, can be driven by a small C-terminal domain in GLUN1. Then that would be potentially an important place to look at. So finally, I'd like to thank you all for listening, and I'd like to thank my lab. Most of the work that was in this talk was done by Ivar Stein here and Deborah Park. And then I'd like to thank my collaborators. We have a great team at UC Davis. Alva was mentioned by Derek already, and then John Gray is an NMDA receptor expert, and Johannes Hell as well. And then my funding sources. And thanks. I'm happy to take any questions.
Fantastic. Thank you so much. Okay, so that was wonderful, both of you. We really appreciate you sharing those insights and kind of flipping iglurs on their heads. If anyone has any questions, feel free to raise your hand or put them in the chat. While people are thinking, I'm going to get us started asking both of you the same question, which is, we're here today to talk about revariance, and something that I'm wondering is the implications from these different studies. Do they say anything about where we could approach treatment options? Like, are there any new targets that we could explore? Anything that may tell us like new ways of I guess, yeah, like targeting these iglurs and how they might be beneficial for individuals with GRI disorders. So we'll start maybe with whoever wants to go first.
I'll bow to the expert and then
Shall I go first?
Yeah.
Oh, actually, okay, sorry. Okay. So I guess the way we've been thinking about it, and somebody actually asked me in the chat just before while you were chatting, Karen, is if you actually start to think about, because if you think about the AMPA receptors, for years we've been sort of talking about NMD receptors being highly regulatable and all this, and they're all those things, right? But the AMPA receptors have this whole combination of things that have to come together in the native system for them to work. There's the pore-forming subunits, the auxiliary subunits, and then you've got alternate splicing, RNA editing. All of that's all coming together, plus all the other stuff that happens to them. What we think, though, is, and we haven't really published all of this yet, but we're kind of working our way through it, is that there's certain parts of brain regions and certain circuits which are particularly vulnerable, right? And so maybe that's the ones to go after. And so one good example of it is recently I was contacted by a mother in Quebec, where I'm in Quebec, and she's got a son who's got a truncated GLUA4 subunit. And GLUA4 is important in the adult brain, although we sometimes play it down, but actually is important in the adult brain, but it's much more selectively expressed. And so you could imagine that you could in a simplistic, simple-minded way, think about targeting those parts of the brain where GLUA4 seems to be important. So, for example, in terms of seizures, it seems to be very important in this thalamocortical circuit. The John Hugenaar, who's out in the West Coast of the US, he had worked out, and there's other people in the cerebellum where we've done a lot of work as well. It's found in the granule cells, and it's also in the vertebral glia. So one could maybe think about trying to target just certain parts of the brain, and that might, for example, control, that might be able to control the epilepsy. And so there's some people thinking about those things as well. There's a paper in bioRxiv with Rick Hugener, who I think is on this call and might be talking about it later on. So there's people coming along with sort of genetics and anti-sense approaches to try and look at that. So that might be one way to do it, or one may be able to come up with small molecule therapies if you just focus on that brain region and really get a deep understanding of what's completely wrong with it, and can you come up with small molecules to fix it, then maybe that's a way forward, right? So that's kind of how we've been thinking about it. We obviously haven't developed anything for the AMPA receptors yet, but we've taken that approach for NMD receptors, and that led us into treatment for Fragile X. Now, we don't have that treatment for Fragile X yet, but it got out of the into the translation patent space and formation of a company. So it's viable to even the business people. So that's one way we've been thinking about it.
Go ahead, Karen. Yeah.
Okay. Yeah, there's also a question in the discussion in the chat, which would you prefer me to do one versus the other, or both? I guess maybe both. I'll try both.
Yeah, that'd be great.
Okay.
Maybe stick to the first question and
Okay.
then
Sounds
feel
good.
free to, yeah.
So I guess for me, one thing that a couple things kind of they'll actually transition perfectly. A couple things stood out to me as I started. Again, since I do most of my studies on on general learning and not as much on glutamate receptors, it was really fun to delve into the literature a lot in the last week on the disease-associated mutations. And I guess what I would say is that one interesting thing to me, again, that I mentioned a little bit during my talk, is the focus has really been on how they're affecting the physiological aspects, and less on how they're affecting the structural aspects of synapses. And I think it will be fun to look at them to see, again, how those aspects are being affected as well. Just because we've seen there's been a lot of separation. If you have, in a normal situation, they're really tightly linked. But in a situation where some change has been made, that can cause the two aspects of plasticity to disassociate and go different ways. And so I think that would be really insightful to try to create new insights. And so, yeah, for me, I definitely am inspired to start looking at some of these disease-associated mutations with our particular aspect of our particular view. And then the question, which is interesting in the chat, is related, and that was essentially, well, what about the C-terminal domain? Is it possible, it says, that loss of the C-terminal domain would still promote the loss of spines? And does the whole receptor tetramer need to be in place without ion flow for this downstream effect? And that's really interesting, just because we have, again, due to the Bonin lab where Quinn came from, we have a little bit of insight, and then we actually adopted their technique just for fun, just to see what it did for us. So essentially, they developed a small peptide. It's not the whole C-terminus, I'd have to say, but they developed a small peptide to show the opposite, actually. If you express the peptide that looks to be quite important for the signaling pathway, it actually acts to block this signaling. It acts to block this signaling pathway, this ion flux independent signaling pathway, by binding away probably whatever binders are required. And so I guess the way to think about it is, I'm going to assume it's likely that that the C-terminus is not, of one of the NMDA receptor subunits, is not going to be enough to drive the turn on of this pathway, because probably you need this whole complex that's assembled by the intact NMDA receptor to turn on the pathway. But turning off the pathway, but we don't know that. It's entirely possible. Again, I won't exclude the possibility that expressing, I've never done it, that expressing a full C-terminal domain of one of the subunits would drive it. But to interrupt it, we certainly can use manipulations at a single C-terminal domain that's critical for allowing the pathway to drive forward. I don't know if that's helpful.
That's great. Yeah, no,
Okay,
that was
cool.
great. Thank you. We have a new question in the chat from Ian Coombs. Are there any expression system experiments that can detect non-inotropic NMDA receptor signaling, or do you need to do it in neurons?
Yeah. So so there's a whole, again, in a longer talk, I'm able to talk about other people's research more, but yeah, there's a whole set of experiments that can be done looking at fret flim changes and conformational changes, and while they're not, so essentially this was stuff that was, experiments that were started by Kim Dorr when she is in the Malino lab. She now has her own lab. And she was looking at fret signals between the two, between the gluin-1 C termini, and I think that's probably what got the Bonin lab to look at that particular aspect of the NMDA receptor as well. But what she showed is that even when the channel is blocked, the C termini of gluin-1 move apart by using fret fluorescence imaging of a red and a green protein that changes the lifetime of the green protein. I guess it's hard to describe in a really short talk, but yeah, you can do a fluorescence assay and show that she showed the C terminus is moving. And she did most of those experiments in a heterologous system, but she also was able to do them, confirm them in neurons. So in that way, but the one issue I would say with that is there's no guarantee that that exact movement is the one that is driving the signaling pathway downstream. We know that so it's correlated. There's a correlation between that movement and the downstream signaling. So that's an assay that could be done in vitro and not in neurons.
Yeah, sorry, rather than type out my next question, I thought I'd just unmute and everything. So in theory, then you could do a screen with GRIN1 variants to see
Absolutely.
if this kind of thing is ongoing. That would be a really interesting series of experiments to do, actually choosing some kind of well-studied ones.
And again, I guess the only caveat that I would say, I think that's super cool. And yeah, I definitely think going down that pathway seems like a good one to do. I should give Kim a call and team up as well. But the only caveat being the one I was saying before is that there's only been a couple experiments that, essentially if you throw in an antibody and that C terminus can't move, it blocks. But what we don't know is that movement could just happen and we could eventually separate that movement from the actual signaling pathway. It just hasn't, but that would be the perfect way to start anyway. So yeah, that would be a great thought.
And then one very, very quick final one. Is there a difference between D-serine and glycine with this property? Because glycine kind of hangs around the whole time, and then if D-serine is more kind of basic, then maybe, yeah. Sorry, I think I should probably know this. This feels like glutamate receptor 101, but
no worries. I would say that unpublished, very surprising major differences that are not published yet. Yeah. There is, and there's some published ones, I think, maybe from, was it Laurent Groch's lab? But we have some, and so does Kim Dorr has some, and John Gray, along with all three of us were working together. We do have some pretty remarkably surprising results that they're
related to the non-ionotropic effects.
Yes,
oh, cool.
Non-ionotropic.
Oh, it was a good question then.
Yeah, that's a great question.
Okay. All right, thank you very much.
Yeah.
Awesome. Another question for Derek. You mentioned GLUA4. We have some gain of function GRIA4 kids in our community. We've been told that these are sunset genes. Are genetic interventions useless or even damaging after childhood? And what we mean by what Lana means by sunset is that they're not as expressed in adulthood as during development.
Okay, maybe I missed it. So it says gain of function. Is that right? They were saying the gain of function. Yeah. So I think that the GRI4, maybe we need to take a closer look at that. I'm not sure if I think we've overplayed that the field has kind of overplayed that the fact that GRI4 disappears during development. Yeah, it's very high in development, it seems. And so is TARP gamma 4, for example. It's very high as well, one of the TARPs. But GLUA4 is absolutely found in the adult brain. It's found in the calyx of health, so it's very important for auditory information. as I mentioned, there's all this beautiful work that's been done by Joe Hogan and a bunch of other people looking at the thalamocortical regulation that gets dysregulated and causes seizures. It's in the cerebellum, it's in the Bergman glia, it's probably in other kind of astrocytes across the brain. And we know from, not from any work that I've done, but from a lot of work that's been done in the hippocampus, that this GLUA4 and a lot of the interneurons, it gives that very, very fast synaptic properties. So the sunset, I forget what the term was he used. I think they're probably very relevant. And as again, you know, we're starting to think about it from an evolutionary perspective. So the way we think about it is things come in, they do a job, but by the time they get into the adult, they're doing other kind of jobs and they're associating with other kind of proteins. And so I guess what I'm advocating for is that if we really want to make advances towards understanding greed disorders and coming up with therapies, then much of our learning and much of our teaching that we've been doing, and I've been doing it as well, so I'm part of that for the last 20, 30 years, 20 years for sure here at McGill, is that we teach undergraduate students that we think is probably not correct, right? It's not completely wrong, but people tend to think about, for AMPA receptors in particular, we tend to think about them purely through the lens of the pore-forming subunits, so GLUE 1, 2, 3, and 4. But we know that's absolutely not true, right? They've all these auxiliary subunits, they change the properties, and it means that some missense mutations, like say for GLUE 4, that would be found all across the brain, it turns out they are much more detrimental in certain circuits just because of how they assemble with different types of auxiliary subunits, and also how they're regulated by flip-flop, RG site. The QR editing is constant all the way. So that's the one thing that's constant. But the RG site is dynamically regulated, and so it's flip-flop. And even you can imagine during Hebbian plasticity and homeostatic scaling, that these properties are probably changing as well. So we need to understand that better. So that means we have to understand new biology first. And that goes hand in hand with understanding greed disorders. We've uncovered new biology. We get some new stuff as well that hopefully we're going to be sending out soon, where things that people had assumed are just not true. But when you put it in the context of greed disorders, it kind of makes total sense, right? So I think this new biology has to be discovered. Karen's kind of doing that as well. And then once we have that, and if we do that in parallel with looking at missense mutations or truncations, then I think in a few years, it won't take too long, in a few years, we'll have a much better understanding of how to go about treating these kids.
Thank you so much. So we have lots of questions coming in the chat. Unfortunately, we only have a couple more minutes. So I think what we'll do in this last minute actually is if each of you could just provide closing thoughts, and like if everyone here could take one thing away, what would you want it to be? And if there's a specific question in the chat that maybe they can email you about, you can also feel free to respond to.
Karen, you go ahead.
Okay. I did want to just, Tyler, just really, because I mean, I guess it's related to closing thoughts. Tyler asked this question in the chat about like the role of mutants, whole grain-related mutant animals, as opposed to just like, can we learn from them? And, you know, I've been doing a lot of thinking about that because one of my approaches has been that I really like looking at small changes, so that single synapses, single neurons, so that I can really decipher, change, to separate out what exactly is happening at this single synapse relative to what are the effects when something has changed in the whole animal. And I would just like to say that I think there's a role for both, because the single synapse is the stuff where, you know, yeah, maybe, or the single neuron, or, you know, one change at a time or one brain region at a time, maybe you're going to get some insights on what effects are happening in those brain regions independent of the other regions. But then, yeah, to develop therapeutics or to try to treat, you know, at the whole animal level, you're certainly going to need to look at whole animals, because that's what's happening in humans. Yeah, so anyways, I guess that's, in human iPSC models, I guess, would be another huge and important road forward in my mind. So using the human patient mutations, but anyways. Derek?
Yeah, so maybe I can finish on a very positive note. I think we've got a great glutamate receptor research community that's been around for a bunch of things. We know each other, and we're prepared to disagree with each other, but it's a great way, and it sort of builds new ideas. So I think that's great. We need to engage more to get translation people involved in what we're interested in. There's obviously great interest from the community of people who are impacted by this. So I think we have all the right ingredients. It's just a question of us making sure we stay on focus and trying to get government to try and focus on giving money in a way which is strategic. And I think that's one of my biggest challenges, is that we just can't get the government to, especially in Canada, to kind of focus on these translational things. But I think we have all the right ingredients, and if we keep moving forward with all the different players, I think we're going to make some important progress. At the moment, I think we're looking at small molecule therapy. I think that's the thing that's going to go forward. But genetics is much closer than I thought. Genetic treatments is much closer than I thought.
Thank you so much. Thank you, Karen and Derek, for being here. That was a fantastic discussion. Thank you all for joining. Our next session will be back in the main room, so you can head over there to hear about clinical trial updates. Thank you again.
Thanks a lot.
Okay, thanks again, everybody. Thanks. Bye.